Hello, welcome back. Today we'll go through the cardiac and smooth muscle. To begin with, the myocardium is made out of cardiomyocytes, and the cardiomyocytes are branched, surrounded by delicate connective tissues that have a lot of capillaries. They're usually mononucleated, that is centrally located within the cell, and these are intrinsically contractile. What does that mean? It means that these muscle cells can continuously contract without without a direct nerve impulse. Like, we're not constantly thinking about our cardiac muscles saying like, hey, contract now, contract now, contract now. Here, as you can see, these are the intercalated discs, and here's the nucleus, and all these like dots around them are either like glycogen droplets or they're mitochondria. The cardiomyocytes are striated, of course, and the actin and myosin filaments are arranged in the sarcomeres. They're not quite as well defined as the skeletal muscles, and numerous mitochondria and lipids and glycogen droplets are found between the myofibrils, like in between these spaces. And they're all connected to each other by intercalated disc, which is analogous to the Z line in skeletal muscle. So here you see, here's the thick filament, myosin thin filaments. Here is the intercalated disc or the Z lines. And the cells together, like look how they're held together. And here's the mitochondria. So the intercalated discs have fascia adherens that anchor the cells together by linking the cell membranes to the actin cytoskeleton. The desmosomes are the spots, and they anchor the cell membrane to the intermediate filament network and keep the cells bound during constant contraction. So since the heart is always contracting, they have like these two attachments. So these cells are quite strongly bound together. And then within the disc, they also have the gap junctions that allow the ions to pass, and a rapid passage of an electrical impulse, so it can like go throughout the entire heart. Mutations and deficiencies in the intercalated disc components can lead to cardiomyopathies and arrhythmias. Okay, so here's the gap junctions, letting the ions come in. Here's the desmosomes. Here's the intercalated disc right here. Here. And the gap junctions are like little spots. Desmosomes are also spots. The fascia adherens is more like a continuous band, really. Let's go back a slide, and the picture seems better in this one. As you can see here, here are the desmosomes, and here's the gap junctions. Okay, so of course there are the myofibrils, which are the actin and the myofilaments, and they separate to pass around the nucleus, which kind of makes it like that kind of like wide, like kind of like a Y shape around the nucleus, leaving a perinuclear clear space filled with organelles and mitochondria. And in the heart, the T tubules are wider than in the skeletal muscle, and there's only one T tubule per sarcomere. And the tubules work to also they also work by like helping the conduction of the impulse to allow the cell to contract, like as a unit. and there's only one per sarcomere, and the sarcoplasmic reticulum does not form a continuous cisterna. Instead, the cardiomyocytes have a dyad instead of a triad. So here's the dyad. There's the T tubule, and here is the cisterna. See, these are only the two of them. While in the skeletal muscle, as you can see here, this is a triad because they have two sides of the cisterna. So like one on this side, one on this side, and here's the T tubule. And in the heart, there's no satellite cells, so there's no regeneration. So after a heart attack, after some kind of trauma to the heart doesn't recover and regenerate the way skeletal muscle does. This is problematic, and because scarring of the heart can happen, and scarring of the heart makes it less stretchy, and that can lead to some kind of like heart failure. In addition, this is why we can recognize whether somebody has had heart attacks in the past. So the ischemia doesn't go away, it forms scars, and us as clinicians, we can take a look actually using like sometimes an x-ray if it's particularly bad, but CTs, like they'll show up. They'll also show up if you go in and look during like surgery. Okay, so the sinoatrial node and the atrioventricular nodes are made out of the modified muscle cells. They have very few myofibrils, but they're modified because they conduct the nerve impulses. In the sinoatrial node, there's the nodal cells or P cells, and they exert the pacemaker function, which is regulated by the autonomous nervous system. So these cells here, the sinoatrial node, they're the ones that tell the heart when to contract. They send the impulse to the left atrium via a tract or a series of fibers called the Bachmann bundle, and excitation reaches the atrioventricular node, and that holds on to the transmission. It delays the transmission to allow the ventricles to fill. So that's why there's that little pause between LUP and DUP, right? So, like I said before, the heart can contract without any innervation, all thanks to like the T tubules and gap junctions. Next is the bundle of His, and that transmits the electrical impulse to the Purkinje fibers. And those fibers have a lot of glycogen because they're myelinated. The myofibrils will lie in the periphery of the Purkinje fibers, and these fibers conduct stimuli 4 times faster than normal cardiac fibers. Why? Because it has to coordinate the contraction between the atria and the ventricle, and the ventricles are much larger, so the impulse has to pass faster to cover a lot more space so that the atria and the ventricle contractions can be synchronized. Artificial pacemakers are implanted for patients suffering with arrhythmias, especially bradycardia, some kind of like heart blocks, and tachycardia. So the pacemaker will be implanted subcutaneously, and then the leads will go into the heart, into the right atrium, into the right ventricle. Okay, so the cardiocytes don't have a stable resting potential. The conductive cells have a series of sodium channels that allow a normal and slow influx of influx of sodium. Sorry. The spontaneous depolarization after the sodium has reached a certain level will open the calcium channels, and then the calcium channels will enter, further depolarizing the cell until the threshold has been reached. So there's like a pre-depolarization, and then when the calcium enters, there'll be like a full depolarization. Once that has reached the threshold, the potassium channels are opened, and the potassium leaves, repolarizing the cell. The calcium also plays a role in the prolonged plateau phase, which we will cover in a lecture regarding purely the heart. And calcium also plays a role in the absolute refractory period, along with removing the inhibition on the heads of the myosin to form the cross bridges. The heart will still perform the same kind of movements as skeletal muscles. So calcium comes in, removes the inhibition, the cross bridge forms, there's the slide, the heart contracts, and then ATP is used to detach. Here, calcium is stored in the sarcoplastic reticulum. As you can see, comes out. Calcium will help it relax, and then contract, and then during relaxation, it's sequestered and stored again in the sarcoplastic reticulum. And see, it goes into like the T tubules. And then, of course, there's the it takes ATP to repolarize the cell. The heart is regulated by the autonomous nervous system, which adjusts the force generated by the muscle cells and the frequency of the heartbeats. The atrial muscle cells are smaller than the ventricular muscle cells, and the atrial muscles in the right atrium will synthesize the atrial natriuretic peptide in response to atrial distension. So once it fills, it will secrete this. It's a peptide hormone, and then elevated levels of ANP, the atrial natriuretic peptide, is synthesized during the hypovolemic states, and this would be an indication of heart failure. This is a pretty important indication of heart failure. So once the clinician has run like a CBC and you see that there's like high levels of ANP, you can automatically assume that the heart right now is going through heart failure and is surrounded by it's too full of blood, and the pericardium might be filled with fluid. So the ANP also stimulates the excretion of water and sodium via the distal convoluted tubule in the kidney, and it also reduces the secretion of renin, aldosterone, and the antidiuretic hormone. And all of it works together to normalize blood pressure and the volume of blood. So here's the cardiomuscular fascicles. Here are the capillary. Here's the cardiac muscle cell. The discs, as you can barely see, but there's nucleus, endomysium. Okay, so next part is smooth muscle. Smooth muscles are in the visceral organs, the blood vessel, parts of the eyes, like the iris, and the erector pili, which are like the tiny muscles that are connected to like the hairs on your body, and it helps it raise the hairs, when you're cold and when you're scared. The smooth muscle are fusiform shaped, which is kind of like spindle shaped. It's surrounded by an external lamina, and they're arranged in bundles or sheets and lie over one another in a staggered fashion. They can be a variety of sizes, like very small, like the erector pili, or large in the uterus. And they are mononucleated, and the nucleuses are centrally located. The sarcoplasm has the actin and myosin myofilaments, and they form longitudinally orientated bundles. I can see, nice. The myofibrils are not aligned, but and also there's no striation. So the smooth muscle cells can also secrete the connective tissue matrix along with large amounts of collagen and elastin. So here is like a multi unit, or this is the visceral type, and this would be like the muscles like here in the eye. This is all like nice and neat. Well, this is kind of like rearranged everywhere. And look how many neurons there are compared to this. Like this one is clearly for something that requires precise movements. Actin myofilaments will insert into like alpha actinin aggregates, also known as focal densities, on the cytoplasmic surface of the cell membrane. And then it extends from the focal densities into the cytoplasm to interact with the myosin filaments. And then the myosin filaments will interact with a second set of actin filaments, which insert into the intracytoplasmatic dense bodies. And then the further actin filaments will extend from those dense bodies to interact with more myosin filaments, and so on and so forth until the last actin filaments of the bundles insert into the attachment plaques. So there's not a lot of endoplasmic reticulum, rough or smooth, and if there are any, it's close to the cytoplasmic surface of the plasma membrane. There are no T tubules, so there's no striations, and the other organelles will accumulate around the poles of the nucleus. And see how it's like relaxed right now. Here's the nucleus, actin, dense bodies, myosin, and they all insert, so they all connect. Here's the sectional view, how they all like interact with each other right here, and adjacent smooth muscle cells are bound together at dense bodies, and those are important because they transmit the contractile forces. And when it's contracted, look how like the edges are scalloped and it's all lumpy and not nicely contracted like the skeletal muscle. So contraction is initiated by an influx of calcium. Calcium will release from the endoplasmic reticulum, and entry from the extracellular space go through the calcium channels. And then the increased cytoplasmic concentration of calcium binds to calmodulin. Calmodulin will activate the myosin light chain kinase to phosphorylate the myosin light chain, and that will allow the binding to actin. And then the phosphorylated myosin light chain kinase will move the inhibition out of the way, allowing like the actin to bind. It's just very similar to skeletal and cardiac contraction, just with the different kinase. And then the energy released from the ATP by the myosin ATPase allows the cycling of the myosin cross bridges with action for contraction, and that just means that ATPase will cleave the ATP, release energy, and allow the heads to detach. So here you go. The dense bodies will be pulled, and then that in turn will pull the dense plaques, forming like an adhesion rings, and they will pull to the cap, and all these cross filaments will all pull together and make like the lumpy surface. So here it is. Calmodulin calcium binds to it, and it activates the myosin kinase, and then ATP is used to activate the myosin. Contraction is transmitted by the intermediate filaments and the focal densities to the network of external lamina, and it allows the entire mass to contract as a unit. The gap junctions allow the spread of excitation between cells, and tone is when the contractile activity determined by the phosphorylation of light chain of myosin is maintained at a low level. So it's similar to skeletal muscle. There's always a tone, why does the tone exist? It exists so that the body doesn't have to like spend more energy to fully contract again. It's kind of like putting your laptop like to sleep by just closing the top, rather than shutting down the computer itself. So a little bit of energy keeps the laptop running. So when you open it up, tap on the mouse a little bit, and your laptop turns on, keeps running. Versus when you shut it off completely, you have to wait forever for it to boot up, and then you have to enter your password. And that's why muscle always has some form of underlying energy, underlying activity. So when it's needed, the body doesn't have to restart everything. Okay, so increased level of CAMP will induce the phosphorylation of myosin and the cell will contract. We already talked about the phosphorylation and how that activates. Let's go through innervation. So smooth muscle is intrinsically contractile, like cardiac muscle, and it will contract in the absence of nervous stimuli. The way it contracts is slow, sustained, and fatigue resistance, and it also responds to autonomous nervous system and hormonal stimuli via CAMP. For example, estrogen will increase CAMP, which leads to uterine contraction. And there's no well-defined neuromuscular junction, unlike the skeletal muscle. And the motor endings of the autonomous axons are not closely associated with the individual smooth muscle fibers. Instead, neurotransmitter activation is fairly diffuse, and it can be stimulated by like histamine, for example, an asthma attack. So it'll be like just one fiber, and it will innervate like a whole lot of smooth muscle cells. It's usually some form of acetylcholine. And look here, this is going to be a bronchial kind of area. And look, it still has the nitrate. Remember, nitrate helps to increase the dilation of vessels. Okay, so the type of stimuli will specify which muscles react. There are two different types of smooth muscles. They're the multi-units, which are functionally independent smooth muscles. For example, they're in the eyes. They have a rich nerve supply and usually innervated by a single nerve terminal. And these are the ones that don't contract spontaneously. There is they are somatic, so an active impulse, an active decision will produce the impulse, and they are responsible for very precise contractions, so like eye movement. Visceral types of smooth muscles have abundant gap junctions and a poor nerve supply. The bundles work together in a syncytial fashion because of the gap junctions, and they contract spontaneously if stretched beyond a certain limit. So like when you swallow, and you know how food travels down the esophagus via like peristalsis, right? So the esophagus needs to be stretched to a certain point so that the muscles know like, hey, there's something here, squeeze. That's why your esophagus doesn't squeeze if you just drink water. Well, that's it for now. Thank you for your attention and good luck studying.